Hello everybody, my name is Thorsten Mumbauer. I'm the CEO of Exivir. Exivir is a clinical stage biotech company developing single domain antibodies with the target to develop an infectious disease platform. I'm very pleased to have an experienced management team with me on this journey. Dominique Tessago is our Chief Medical Officer. Dominique has long experience in developing nanobodies. She had been CMO at Ablynx in the days when they designed the phase 3 trial for caplacizumab. Fiona Monceau is our Chief Operating Officer, also 20 years plus in biotech and pharma experience, mainly commercial roles, and Fiona joined us from UCB Venture Partners back into the operational role here at Exivir. Exivir is a spin-off of the VIB in Flanders. We have a long-lasting R&D collaboration with the Institute, and the key scientists and developer of the technology, Professor Salens and Nico Kalawarts, are also as consultants with the company. The nanobody platform technology offers very nice solutions for treatments of infectious diseases with highest medical need or pandemic preparedness. Exivir is going to develop the platform in both directions. We are convinced that we have proven the utility of the platform for pandemic preparedness by developing our lead candidate from bench, from the lab to patient in less than a year. Here's the platform technology in a little bit more details. So nanobodies are derived from camelids, in our case from llamas. The llama will be immunized by the target antigen, in our case a spike protein, in our case of our lead product, the spike protein. You extract the library, you get out the nanobodies, you identify the epitope of binding, and then you optimize your candidate up to the lead candidate. As the nanobodies are very small, they can target very special epitopes on your target antigen, and you can do interesting mix and match combinations. In case of our lead candidate, we have a homologous binary molecule where we fused 2 VHHs to 1 Fc backbone. We are working on second generation and new molecules where we have proven that you also can work with heterologous molecules. So in that case, we have identified by boosting the llama again and further activation and optimizing the immune response, a new very exciting epitopes on the spike protein, and we have fused 2 different VHHs to 1 Fc, and we have similar and even higher potency in these molecules, which mimic the concept of having 2 antibodies in a mixed, as some of our competitors in COVID-19 do. The low dose, the high potency of the molecule also offers the possibility to move into subcutaneous application and even into FC. Actually, we are manufacturing in CHO cells, standard process, but the small size of the nanobodies also allows manufacturing in yeast. The lead candidate. It's a highly potent, broadly neutralizing SARS-CoV-2 nanobody. It was originally generated by immunizing the llama against SARS-CoV-1. As soon as the SARS-CoV-2 sequence became public in February last year, the scientists set up a screening assay and found 100% cross-reactive binders against SARS-CoV-2. And that led to the development of our lead candidate. We are binding to a very high conserved region on the spike protein, which is essential for the virus to enter the cell. There is a couple of very key differentiation factors for our nanobody. The first one is the small format, so it's just 80 kilodaltons, so roughly half of the size of a human antibody. That allows a much better tissue penetration into the body, and if you're using the same dose, you have double of the molecules in the body of the patient. As I said, we have a very highly conserved epitope. It's conserved over the Sarbecovirus family, which has also proven the importance for the virus. There is very little mutation pressure on the virus, and so we do not see any escape variants in any of the variants of concern so far. Very important differentiation factor. On top of that, we have silenced the FC molecule, the FC part of the molecule. We want to avoid any exacerbation of the disease in infected patients. Therefore, a silenced FC is a key safety feature from our molecule, which differentiates us within the COVID-19 therapeutic antibodies. On the left side, we see a graph. The green part, the upper green part, is the ACE2 receptor, the binding site. The grayish is the spike protein, and the blue ribbon on the right is our lead candidate with the binding epitope. We have also visualized here in red and yellow the mutation hotspots of the variants of concerns. You see there the binding domain, our binding domain, is clearly differentiated from the mutation hotspots. And as I said before, we do not see any loss of efficacy or potency against the variants of concern. We are having a dual mode of action for this molecule. Together with the FC fused to the nanobody, you would see that on one hand we do the same steric hindrance of the binding of the spike to the ACE2 receptor as the other antibodies, which have the binding domain more in the upper part of the RBD. And on top of that, on the right side, you see the shedding of the S1 subunit, which is the part of the spike protein. We are using Raji cells here, which express the spike protein on the surface, incubate with our antibody, and see the shedding of the S1, which means that the binding of the antibody destabilizes the spike protein and sheds off the molecule from the cell surface. Some in vitro data. So standard model in SARS-CoV-2 is the Sarin golden hamster. On the left part, in green, we see the therapeutic treatments. In red, orange, we see the prophylactic ones. Blue is the control. This is infected virus particles 4 days after infection. In green, on the high dose, we see full protection 4 days after the infection, almost full with a low dose. In red and orange or yellow, we see the prophylactic usage, where the animals have been treated 1 day before the infection, and we see full protection also after 4 days. This is an outlier. I mentioned before that we do not see any loss of potency against the variants of concerns. Here we see the neutralization data against alpha, beta, gamma, and delta variants, and we see almost no difference against any of the variants of concern. So we have a highly conserved epitope and no indication that there's any viral escape. We are not alone in this. It's a very busy, very competitive environment, but the positioning of our molecule, I believe, makes it special again. So we are looking in our first trial into hospitalized patients. What we see here in the comparison with all our competitors, that most of these molecules are starting in diagnosed or post-exposure or even in prophylactics. And there is not a lot of activity in hospitalized patients. We believe that hospitalized patients are having the highest medical need to avoid disease progression and even going into ICU or death. We're starting our development, as I said, in hospitalized patients, mild to moderate disease. We then, in the next step of our development, want to move into the high-risk population, into the most vulnerable subgroups, or specifically into the immune-compromised patients. The long term is also to avoid long COVID, where we also believe that the better biodistribution of our molecule can make a big difference. But there, we definitively need additional data over time. On the left part, try to indicate the market size in this specific patient group. We have recently seen the sales numbers from some of the competitors, so it is a big market opportunity. Here's the clinical development plan. So we are actually running a phase 1A in healthy subjects, and we have an open phase 1B in patients, hospitalized patients. We have infused the first group in the phase 1A, 10 subjects, no side effects. We will infuse the second cohort, the middle dose, this week. We have also included the first 2 sentinel patients in the 1B, and that means that the phase 1B is open now in 3 sites in Belgium, 2 sites in Italy, and we will add 1 in Portugal very soon. We will have interim data around November time. We will have an IDMC to make a decision on the dose. That will lead to an opening of a phase 2 international, which will include sites in the US, in Mexico, Brazil, and in South Africa. We should have data around Q2 next year, and that will lead to a filing of a marketing authorization mid-summer next year, and we'll hope to get a conditional approval in the winter 22. Exivir was focusing in our first year on the development of the lead candidate XVR011. We are clinical stage now. We are filing for the MMA. We are in parallel working on the manufacturing process. We have booked commercial slots, 6 commercial slots at a European-based CDMO. We hope to get a conditional approval end 22 and be a commercial stage company 23. In parallel, we are working on our pipeline. We've worked on the biology of the platform over the year. We are in the process of identifying our lead indications. We'll open a lab at the incubator at the VIB beginning of next year, and so we hope to have the lead candidate selected end of next year and start with the CMC process. Today, Exivir had raised a total of 24 million in a Series A. We have added a 3 million Vlio grant to that, and we are in the late stage of signature to get another 3.5 million grant in Belgium and a 10 million grant from the EU. To finance all of our future plans, specifically the consistency development lots, the development of a sub-Q in a lifecycle management, a potential phase 3 confirmatory trial after the conditional approval, we want to raise additional 100 million euros. We want to do that in a 2-step process, so 50 million in an accelerated way forward again, hopefully to be closed before year end, and an additional 50 million plus over the next year. That might be a Series C. We will also be looking at the public market to monitor the development and possibilities there. Please feel free to reach out to us during this conference. Would be very pleased to give you a more detailed presentation and to answer all your questions you might have. Thanks a lot. Looking forward to hearing from you.